Today, I will speak about the reaction of plant cells under hyperosmotic conditions. But to begin, why we can speak about osmosis in a plant cell? Osmosis is a phenomenon involving the passive movement of matter across a semi-permeable membrane that separates two solutions with different solute concentrations. Because the membrane is semi-permeable, solvent molecule can pass through it but not solute molecule. As a result, the solvent molecule move from the less concentrated solution to the more concentrated one until there is an equilibrium of the concentration in the two media. Plant cells are subject to this effect due to their structures. The main elements of the cell involved are the plasma membrane, the vacuole and organelle, and the cell wall, the outer border of the plant cell. If the solute concentration in the vacuole and outside the cell are different, the vacuole will be more or less filled with water due to osmosis, which went to have the equilibrium of the medium. This leads to a pressure from the plasma membrane on the cell wall called Tugger pressure. In normal conditions, the cell wall strength and the Tugger pressure strength are in balance. However, for instance, in the case of water stress due to draw or salt stress, plant cell can be in a hyperosmotic environment. The concentration of solute outside the cell becomes higher than inside the cell, causing water to flow out of the cell and Tugger pressure to decrease, breaking the establishing balance of forces. More precisely, the cell will react in two ways. If solutes from outside the cell are allowed to pass through the cell wall but not the plasma membrane, the protoplasm will contract due to osmosis and the plasma membrane will separate from the cell wall while remaining attached to it. This is called plasmolysis. This state is irreversible at first, but if it persists for too long or as a result of excessive osmotic pressures, the plasma membrane may completely separate from the cell wall, becoming irreversible and possibly resulting in cell death. Plasmolysis rarely occurs in nature and is mostly used in laboratories. However, most of the time the solute outside the cell cannot pass the cell wall, so the whole cell will contract. This is called cytolysis. But if the osmotic pressure becomes such that the Tugger pressure is negative, the cell can also end up dying.